Hello, and thanks for joining us today for the latest Technology Networks webinar, Subcellular Fractionation and Enrichment Strategies for Improved Mass Spectrometry Analysis. I'm Jack, Editor for Technology Networks, and I'm here to moderate the event. I'm pleased to have Barbara Kabord and Ryan Baumgarten joining us today as your presenters. Barbara is a Senior R&D Manager for Protein Biology at Thermo Fisher Scientific in Rockford, Illinois. After receiving her PhD in biochemistry from the Medical College of Wisconsin, she performed a postdoc fellowship at the Pennsylvania State University under the direction of Stephen Benkovich. Barbara has over 20 years of industry experience and currently leads a team of R&D scientists in new product development in the areas of protein interactions, affinity chromatography, and protein sample preparation. Ryan completed his PhD at Stanford University under Dr. Carleen Simprich, where he worked on biochemical characterization of the checkpoint kinase ATR DNA binding activity and signaling in UV-sensitive human cell lines. After a brief postdoc in the Simprich lab, Ryan joined Thermo Fisher Scientific as a research scientist for their protein function group, where he was the technical leader responsible for developing over 30 new products. In 2009, Dr. Baumgarten joined a new mass spectrometry reagent group under Dr. John Rogers, where he is currently a senior staff scientist, responsible for the development of products for the proteomics reagent market segment. Warm welcome to you, Barbara and Ryan. Following the webinar, we will have a Q&A session, and we'd welcome any questions that you may have. To ask a question, you should enter your question into the box on the left-hand side and click Submit. We will answer as many as possible during the time available. Any questions we don't have time to get to, we'll be sure you are contacted offline with an answer. If you experience any technical issues, please use the question box to request support. Please remember you can ask questions at any time, even when watching the webinar on demand. I will now hand over to Barbara and Ryan.
All right, thank you for that introduction. My name is Ryan Baumgarten, as I was just introduced. First, I'd like to begin with an overview of today's presentation. I will be presenting a brief introduction to some of the challenges that are faced by proteomic researchers and biologists using mass spectrometry to study complex protein samples. Then my colleague, Barb Kabord, will present on different strategies to overcome some of these challenges. Three of the strategies that we will cover in depth include subcellular protein fractionation, integral plasma and organelle membrane protein enrichment, and immunoprecipitation mass spectrometry, or IPMS, which is a powerful immunoselection technique for targeted mass spectrometry applications. One of the major challenges faced by biologists doing mass spectrometry-based proteomics is that a majority of the proteomics researchers are focused on protein identification and characterization. As such, the instruments and methods available for proteomic analyses using mass spectrometry can most readily provide information about a protein sequence and its various isoforms, including a wide variety of post-translational modifications. Traditional methods used to prepare protein samples for MS analysis requires enzymatic digestion of proteins to peptides. This approach, commonly referred to as bottom-up proteomics, relies on peptides for database searching and subsequent protein identification. However, much of the original protein biological context is lost during this type of sample preparation. Proteins are typically extracted from whole cell lysates using denaturing buffers, which results in loss of information regarding protein localization and protein-protein interactions. Although protein expression data can be inferred from quantifying peptides, this information alone is not sufficient to understand the biological pathways that are active in a cell. Another challenge faced by biologists doing mass spectrometry experiments is that the list of proteins identified by global discovery proteomics is not complete. An example of how this is shown on the left, where a typical discovery proteomics dataset can readily identify and quantify thousands of proteins across multiple sample types or conditions. However, when mapped on the known biological signaling pathways, many of the known lower abundant proteins are not detected. In addition, many of the PTMs that have known function in the literature may not be identified by MS due to low protein sequence coverage. This lack of comprehensiveness results in major gaps that provide an incomplete analysis of the proteome, making it difficult to test hypotheses regarding specific proteins or to validate data observed with immunoassays such as Western blot or ELISA. Finally, proteome complexity is still a major challenge in proteomic analysis. Even using extensive, high-resolution analytical peptide separations, modern LC-MS instruments can only reproducibly identify roughly 2/3 of the proteins expressed in a mammalian cell. And of the proteins identified, the sequence coverage can range from 1 to 70% depending on the protein abundance in the sample. So if a biologist is interested in studying a particular protein complex, or signaling pathway, they need to employ strategies at the protein level to help increase the odds of finding the protein they are interested in. There are a lot of techniques that can be used to reduce the complexities of protein samples. Highly abundant proteins are easily identified with simple protein digestion without further sample preparation. However, medium and low abundance proteins require additional sample preparation to identify them in complex samples. Some of these techniques include abundant protein depletion, protein fractionation by size, charge, or cellular location, or protein enrichment using affinity chromatography techniques. The rest of the presentation will focus on different strategies to enrich low abundant proteins to help increase the odds of identifying relevant proteins of interest, in addition to maintaining some of the biological context, such as the protein's locations, interactions, and modifications. Now I'll turn it over to my colleague Barb.
Thanks, Ryan. There are many ways. I'll start with talking about subcellular fractionation by differential detergent extraction. There are many ways to fractionate cells, ranging from the tried and true density gradients and differential centrifugation protocols to differential detergent extraction to more sophisticated enrichment of specific cellular organelles. Each method has its advantages and its challenges. Density gradients require the use of an ultracentrifuge and considerable time and care to prepare, spin, and recover separated protein fractions from the gradients. While this method clearly gives good results in terms of resolution, it is time-consuming and not amenable to higher throughput. Differential detergent fractionation also provides some biological context, but it is much faster and it's easier to process multiple samples and scale up if necessary. While the yield is good, we need to deal with removing multiple detergents and salts from the various subcellular fractions before proceeding with mass spec analyses. Lastly, the affinity enrichment approach utilizes immobilized antibodies to pull out specific organelles. This strategy is only as good as the antibodies used and is usually limited to very small-scale preparations, as the reagents can be quite expensive. From a practical standpoint, the protein yield, speed, and ease of use of the differential detergent fractionation approach provide significant advantages despite the later need for detergent cleanup steps. Shown here is a schematic of a stepwise detergent fractionation method that we've optimized to give relatively clean separation of major cellular compartments. The first step is to isolate the cell's cytosolic contents by introducing a mild detergent that essentially punches holes in the plasma membrane, allowing cytosolic proteins to diffuse out of the permeabilized cells. A second detergent is utilized to solubilize most membrane proteins, including those of some of the major organelles, such as mitochondria and endoplasmic reticulum. Soluble nuclear proteins are then extracted by the high salt present in the nuclear extraction buffer. Proteins that are more tightly associated with chromatin are released by introducing micrococcal nuclease to fragment the chromatin into smaller, more soluble pieces. The final fraction, or pellet, is solubilized with a much stronger denaturing detergent and consists largely of cytoskeletal proteins and those proteins not effectively solubilized in earlier steps. This subcellular fractionation protocol is also frequently utilized to verify cellular location of specific proteins and to monitor movement of proteins between cellular compartments, such as movement of proteins from cytosol into the nucleus. To show how effectively proteins are fractionated with this protocol, Western blot analyses were performed, probing for specific marker proteins that are known to be found in each respective fraction. Starting with the first panel, HSP90, a protein known to be located primarily in the cytoplasm, is shown to be largely found here in this first fraction. The membrane fraction contains plasma membrane proteins such as EGFR, as well as proteins found in mitochondria like VDAC and the endoplasmic reticulum. The example here is calreticulin. The transcription factor SP1 is found almost exclusively in the soluble nuclear fraction, as is a significant amount of the DNA-modifying enzyme HDAC2, with the remainder found associated with the chromatin. Histone H3, integral to the structure of chromatin, is found exclusively in the chromatin-bound fraction. Cytoskeletal proteins, vimentin and cytokeratin, are found primarily in the last insoluble pellet fraction. Now, when we perform mass spec analyses and looked at all of the proteins isolated in each subcell fraction, the sum of all the proteins, depicted by the gray bars, and peptides, which are the red bars, identified in each fraction exceeds the number identified in a parallel whole cell lysate by about 2.5-fold. So more than 40,000 unique peptides corresponding to over 5,000 protein groups were identified, providing a much deeper dive into the total cell proteome than what can be achieved without fractionation or enrichment. If we examine the proteins identified in each fraction and look for the location of the same marker proteins tracked earlier by Western blot, we see that the mass spec data mirrors the previous Western blot data. The red bars show the primary location of the marker proteins, laid out sequentially per the protocol schematic shown at the bottom of the slide, while the gray bars show other subcellular locations where a lesser portion of these proteins were also found. In the case of HSP90, which was shown by Western to be in the cytosolic fraction, the red bar indicates the majority of that protein found in this first fraction, while the lesser gray bars show a smaller population in the membrane or second fraction. A more quantitative approach to assess the relative enrichment achieved by subcellular fractionation employs the use of tandem mass tags. TMT tags are isobaric labels consisting of an amine reactive functionality and a mass reporter spanned by a linker that is cleaved specifically at the designated location, shown in green, in the mass spec. Each complete TMT tag has the same overall mass, but differ on the specific location of incorporated heavy atoms, denoted by asterisks in the structures on the right half of the slide. Thus, TMT-labeled peptides behave identically in the mass spec, but as the linkers are cleaved during MS-MS fragmentation, they liberate mass reporter ions of different masses. The relative quantity of each reporter ion is then compared. This schematic shows the way we use these TMT tags in our experimental workflow. Typically, different samples are labeled with a different TMT tag. In this case, the 5 subcellular fractions and the control, which is a whole cell lysate, were run into an STS-Page gel to remove salts and detergents, reduced, alkylated, and digested with trypsin, and the resulting peptide fragments were each labeled with a different TMT 6-plex tag. Labeled samples were combined, fractionated by high pH reverse phase chromatography, cleaned up, and analyzed by mass spec, while the same peptides from each sample resolve identically in the first phase, shown in the bottom left profile. Following fragmentation of the TMT labels, the reporter ions resolve by their respective mass, and their relative intensities can be compared. Quantification of 2 representative marker proteins, HSP90 and histone H3, is shown here, and their subcellular partitioning correlates well to the previously shown Western blot and label-free mass spec data. HSP90 is enriched in the cytosolic fraction, histone H3 in the chromatin-associated fraction. But in addition to the proteins whose behavior and localization we already know or predict, relative quantification of TMT labels allows us to confirm enrichment of other protein groups that may not be as well characterized. This table shows several protein groups described by their gene ontology, or GO annotations, that are significantly enriched in particular subcellular fractions. Some, such as brisk deubiquitinating proteins in the cytosol and transcription repressor complex in the soluble nuclear fraction, would be predicted to be localized in the indicated compartments, while others, such as mitochondrial proteins in the membrane fraction and some of the lipid raft-associated proteins in the cytoskeletal pellet, associate into these subcellular fractions based on their detergent solubility. To summarize this portion of the talk, separating proteins according to their subcellular location reduces sample complexity, resulting in a higher number of protein identifications while maintaining some biological context. This translates into deeper proteome coverage and more precise assignment of protein localization. Protein expression or enrichment levels determined by TMT ion reporters correlated really well with other methodology, such as label-free mass spec and Western blot analysis. In addition, gene ontology annotation analysis showed significant protein enrichment of predicted marker proteins and enabled assignment of other protein functional groups to specific subcellular fractions based on their detergent solubility. So I'd like to move to the second enrichment strategy, which is membrane protein solubilization. This will bear some similarity to the previous subcell fractionation approach, except will focus on isolating one specific, very difficult to isolate fraction, membrane proteins. Membrane proteins can be isolated by both mechanical and chemical methods. Traditional mechanical methods result in membrane proteins being retained in native lipid bilayer vesicles, bearing some resemblance to their native environment. This is the major advantage of this approach. However, these mechanical disruption methods are tedious, making it difficult to process multiple samples simultaneously. Alternatively, chemical disruption methods are usually much easier to perform. The use of detergents to disrupt cell membranes is efficient and quick, but the proteins are now in a detergent environment rather than associated with their native lipids. Non-detergent methods are generally less efficient at solubilizing membrane proteins and are typically used to separate peripheral from integral membrane proteins. Although using detergents results in the highest yield of soluble membrane proteins, detergents are very incompatible with mass spec workflows, and care must be used to completely remove them prior to LC-MS. I'll have a slide later in the talk that shows the problems detergents create in mass spec profiles and how they compromise assay sensitivity. The good news is that there are multiple ways to remove detergents. The differential detergent solubilization strategy from the MEMPR plus membrane protein All right, sorry, a little bit difficulty. Okay, so the detergent solubilization strategy from the MEMPR plus membrane protein extraction kit, shown here on the right, has some similarities to the subcellular fractionation protocol, but focuses in on the two steps used to isolate the membrane protein fraction. The protocol is relatively quick, and it can be accomplished in less than an hour, and involves just two quick centrifugation steps to successfully separate membrane proteins from cytosolic proteins and recover an enriched soluble membrane protein sample. Western blot analyses show a good, clean separation of the membrane proteins, pancadherin from plasma membrane, cytochrome oxidase 4 from the mitochondrial membrane, in lane M, while the cytosolic protein HSP90 can be found in lane C. Further Western blot analyses of soluble membrane protein fractions from 3 different cell lines show that the detergent-based extraction method of MEMPR Plus, compared to protocols using non-detergent containing buffers and sodium carbonate buffers, show that the multi-spanning integral membrane proteins, sodium potassium ATPase and ATP translocase 3, are more efficiently extracted with detergent, although complete solubilization is still quite difficult. For example, if you look at the fractionation of sodium potassium ATPase from Hela cells in the upper left blot, you can see that there is slightly less than half of the protein found in the soluble membrane fraction, lane M, with the remainder in the insoluble pellet, lane P, and nothing in the cytosolic fraction, lane C. In contrast, none of the sodium potassium ATPase is solubilized by the first non-detergent base buffer, all in lane P, and the second solubilization buffer lacking detergent shows the same relative distribution as MEMPR plus lanes, but has a much lower overall signal or yield. Sodium carbonate buffer was able to solubilize little to no sodium potassium ATPase, and what was extracted was found in the cytoplasmic fraction, lane C. The other cell line shows similar trends, with the MEMPR plus buffers consistently showing higher and better membrane protein fractionation. Modifying the salt concentration in the MEMPR plus solubilization buffer, changing from hypotonic to isotonic, increases the relative extraction efficiency for both the sodium potassium ATPase and ATP translocase 3 proteins, driving more of these multi-spanning membrane proteins from the insoluble pellet into the soluble detergent vesicles, lane M. Improved fractionation ranges from 10 to 30% additional protein. So we wanted to look at the total protein population isolated by the various membrane protein isolation methods indicated in the previous slides by mass spec. As mentioned earlier, samples containing detergents are incompatible with mass spec and require cleanup to remove these detergents. Detergents in MEMPR Plus membrane fractions and EMPER whole cell lysate samples were removed by simple methanol chloroform water extraction, then reduced, alkylated, and digested with trypsin. Looking at the total number of protein IDs, the detergent-based MEMPR Plus extraction methodology resulted in a similar number of total proteins identified compared to a whole cell lysate reagent such as EMPER, but a significantly higher number, roughly 3 times more, integral membrane proteins identified in MEMPR Plus samples. The number of integral membrane proteins found in samples prepared without detergents, such as buffer 1, 2, and sodium carbonate, is substantially lower than the detergent-based protocol, and interestingly enough, even lower than the whole cell lysate prep shown in the first column. Searching the mass spec data for specific types of membrane proteins shows that our detergent extraction method, in the red bars, significantly enriches peripheral and integral membrane proteins compared to non-detergent methods and to the whole cell lysate preparation, such as using EMPER. Peripheral and integral membrane proteins ranging from 1 to 4 transmembrane domains had higher sequence coverage with MEMPR Plus samples compared to these other commercial and literature methods. As do proteins with higher number of transmembrane domains, such as the proteins shown here, which range from 6 to 12 transmembrane bands. These proteins are much more difficult to extract, but are shown to be significantly present in the MEMPR Plus solubilized fraction. While MEMPR Plus solubilization buffer efficiently extracts multi-spanning membrane proteins, the extraction and solubilization process does not necessarily disrupt protein interactions found in some multi-subunit membrane proteins. An IP reaction was performed with MEMPR Plus membrane fraction isolated from Hela cells using an antibody against the beta subunit of the sodium-potassium ATPase, thermoscientific pierced protein AG magnetic beads, and MS-compatible buffers. The subsequent analysis of the immunoprecipitated fraction identified peptides from the beta subunit as expected, but also identified peptides from the alpha subunit, indicating a successful co-IP of these 2 subunits. To summarize this section, multi-spanning membrane proteins are efficiently extracted using the MEMPR Plus membrane protein extraction kit and can be even further improved with the addition of salt. A chloroform methanol water cleanup of the extracted samples removes the detergents used and improves the ID and sequence coverage of membrane proteins by LCMS. Lastly, the non-denaturing detergent used to solubilize membrane proteins does not disrupt membrane protein complexes based on our ability to IP alpha-beta subunits of the sodium-potassium ATPase. The final enrichment strategy I would like to discuss is immunoprecipitation upstream of mass spectrometry. While the previous 2 sections of this talk focused on more general fractionation strategies, IPMS is a more specific or targeted enrichment approach. As the name implies, IPMS involves immunopurifying a specific protein out of a complex lysate. Immunoprecipitation provides a high degree of enrichment and enhanced sensitivity, while mass spec detection enables specific identification of the proteins present, provides another degree of selectivity in its own right, and is amenable to multiplexing. This targeted approach enables you to simultaneously look not only at protein expression or the amount of a particular protein that is present, but also look at any post-translational modifications in that protein or identify any potential interacting proteins that may co-immunoprecipitate. The end goal was to develop an optimized method that would decrease the total number of proteins present and minimize background while maintaining compatibility with downstream mass spec sample prep. Accomplishing all of this will enable a higher number of target peptides to be identified, lending higher confidence to the protein identifications. . The typical IPMS workflow is shown here. Immunoprecipitation can be performed on a variety of sample types, including cell culture, tissue lysates, and plasma or serum. Following the IP reaction, the recovered sample can be reduced, alkylated, and digested either in gel or in solution, cleaned up or desalted using C18 reverse phase, and analyzed by mass spec either in a pure discovery mode to identify proteins and post-translational modifications, or in a more targeted approach that involves spiking in a heavy peptide for absolute quantitation. Western blots are still part of the mass spec workflow, as we found that many users still prefer to corroborate mass spec data with Western blot data. Multiple immunoprecipitation protocols exist, but we prefer the indirect approaches illustrated here that incubate antibody with a sample overnight, followed by introduction of protein AG magnetic beads, or, if the antibody is biotinylated, streptavidin magnetic beads. The overnight antigen capture maximizes sensitivity of the IP, which is needed to detect most low abundance proteins. Following capture of the immune complex on the magnetic beads, the beads are washed thoroughly to remove nonspecific background proteins, and the antigen is eluted in a buffer that is mass spec compatible, enabling direct processing within solution digestion protocols, which maximize sample recovery and sensitivity of the IP. Immunoprecipitation with streptavidin magnetic beads requires biotinylation of your protein, or of your antibody, before use, but has the advantage of minimizing the amount of antibody that co-elutes in your sample, which can, depending on the amount of antibody present, overwhelm the mass spec and diminish sensitivity. In addition, streptavidin beads are best used with serum or plasma samples that contain significant quantities of endogenous antibodies that can interfere with the IP if protein AG were used. The protein AG beads, however, are versatile in that they bind a variety of antibody isotypes, and their best use is when antibody is in short supply and contains carrier protein that would interfere with antibody biotinylation using the streptavidin approach. As mentioned in an introductory slide, IP dramatically reduces sample complexity, thereby boosting sensitivity. The mass spec profile on the left shows data from a crude unfractionated cell lysate, with the red arrow showing the peak location for the protein of interest, in this case, the EGF receptor. On the right, the mass spec profile shows that the peak correlating to EGFR is significantly enhanced and much easier to identify following immunoprecipitation. As I mentioned earlier during the membrane protein enrichment and subcellular fractionation portions of this talk, residual detergents from lysis buffers or wash buffers can interfere dramatically with mass spec detection. Here, the top profiles show how residual detergent can follow the LC column, profile shown on the left, and contaminate the entire MS profile, shown on the right, increasing noise and decreasing sensitivity. The lower portion of the slide shows an LC profile of an IP elution obtained using our IP protocol and mass spec compatible kit buffers. As you can see, there are no residual detergent peaks to complicate the analysis, resulting in a profile with a significantly improved signal-to-noise ratio, which enables the detection of the relevant proteins. To illustrate the power of the IPMS strategy, we chose to target the insulin-like growth factor receptor PI3 kinase AKT mTOR pathway as a model system. This pathway is normally involved in cell growth and survival control, but is also involved in tumor formation and maintenance of malignant cells. Most signaling proteins in pathways such as this one are not found in high copy number, and monitoring changes in their expression and in their phosphorylation status is difficult. However, before performing immunoprecipitations of several proteins critical to this pathway, we use SILAC, stable isotope labeling with amino acids and cell culture, to metabolically label proteins for mass spectrometry-based proteomic analysis. HCT116 cells were grown either in the presence of light or normal lysine and arginine as the control, or in media containing heavy amino acids for the treated or experimental sample. Cells were serum starved for 24 hours before the heavy cultures were treated with IGF. Heavy and light cells were harvested, lysed, and combined into one sample before immunoprecipitating the proteins of interest. The resulting sample was then prepared for mass spec using in-solution trypsin digestion. Several different immunoprecipitation reactions were performed using antibodies against key proteins in this AKT-mTOR signaling pathway. That includes a variety of receptors, adapter proteins, and kinases, including some that were against phosphorylated proteins. 11 total targets were identified in this initial study, with significant enrichment of phosphorylated isoforms of both AKT1 and 2 and GSK3 alpha and beta subunits in IGF-treated cells. The upregulation of these proteins indicated by the elevated heavy-to-light ratios. Remember, the heavy cultures were treated, the light were controls or untreated. Slight increases in phospho-mTOR and phospho-P70S6 kinase were also observed. Some targets, such as PI3 kinase, P10, and AKT isoforms, are present at low to sub-nanogram levels, yet these proteins were enriched sufficiently by immunoprecipitation for us to be able to identify a relatively high number of unique peptides from each. In most cases, we were able to identify or see the physiologically relevant phosphopeptides in the profiles as well. Because many researchers prefer to perform Western blots in parallel to confirm and support mass spec data, we show here that Western blot analyses of IP eluates of control and IGF-stimulated lysates also show more phospho-AKT and phospho-mTOR upon IGF stimulation in the lower blots, while the total proteins levels remains the same, shown in the upper blots. As shown in the previous summary table of the IP enrichment data, numerous low copy number signaling proteins were identified, most identified by multiple peptides. This slide shows the excellent sequence coverage obtained for AKT, indicated by the yellow highlighted amino acids, as well as the identification of phosphoserine 473 on the last peptide. Detailed mass spectrum on the lower portion of the slide confirms this phosphorylation site. IP facilitates the identification of low abundance targets, but the mass spec was also able to identify other proteins in the eluate, many that are confirmed or potentially interacting proteins, denoted by the red asterisk in the pathway schematic on the right. For example, PI3 kinase interaction with IRS1 increased significantly upon IGF stimulation, shown by the heavy-to-light ratios greater than 1. Other proteins that were detected in the phospho-mTOR IP, such as GBL, Richter, and SYN1, are known interactors of mTOR. Because mass spectrometry can accurately identify proteins based on mass, it is a better detection tool for discovery experiments like co-IP than Western blots, which rely on antibodies and therefore some preconceived ideas of what to expect. The real power of IP enrichment is driven home in this last summary table. The majority of the low abundance signaling proteins identified by IP mass spec, receptors, PI3 kinase, P10, and AKT isoforms, could not be detected without immunoenrichment, as shown by the failure to detect these same proteins in a whole cell lysate prepped and injected directly into the mass spec. Even targeted quantification by SRM, or selected reaction monitoring, which tunes the instrument into the precise known location of the relevant peptide in the mass spec profile, could not detect some of these low abundant proteins, at least those tested, without prior IP enrichment. To summarize this last section of the talk, IP enrichment is extremely important in facilitating mass spec detection of low abundant proteins. SILAC combined with IP enables relative quantification of protein levels between treated and control samples, and post-translational modifications were identified using phospho-specific antibodies for enrichment, followed by mass spec analysis. In addition, the amount of potential protein interaction information obtained by IPMS is very powerful and more easily obtained than through traditional Western blot analysis. To conclude the webinar, I would like to reiterate some of the benefits of protein enrichment, such as the subcellular fractionation, the membrane protein enrichment, and immunoprecipitation upstream of mass spectrometry. All of these methods, in their own way, facilitate the detection of more difficult, low abundance proteins by increasing sensitivity and eliminating some of the most of the background proteins. Subcellular fractionation, in addition to simplifying the proteome, facilitates translocation studies of signaling or regulatory proteins that move between cellular compartments to exert their effects. Membrane protein enrichment enables increased detection of very difficult protein fraction, especially multi-spanning transmembrane proteins. And IP enrichment dramatically increases the sensitivity of mass spec detection, enabling identification and monitoring of PTMs, and is extremely valuable in identifying protein interactions. Finally, I would like to acknowledge all of the talented scientists in the protein analysis group at Thermo Fisher Scientific who generated all these data, and would like to thank you for attending this webinar. At this point, Ryan and I would be happy to entertain any questions you may have.
Thank you very much, Barbara and Ryan. I'm sure you will all agree that there were some excellent points raised during the presentation, and we've had a number of questions, so we'll move straight on to these if it's okay. The first question that's been sent in to us is, which of the subcellular fractionation techniques would I use to enrich proteins from specific organelles?
That's a very good question. So I think based on the stuff that Barb just presented, with the differential detergent method, unfortunately you do, with the membrane fraction, does contain both plasma membrane as well as membrane proteins from the different organelles. So if you're interested in enriching proteins from a specific organelle, the sucrose density gradient sedimentation would be the preferred method because it does allow you to separate lysosomes from peroxisomes and mitochondria.
Thank you, Ryan. We'll move straight on to the next question. Somebody has asked, how are detergents removed after subcellular fractionation using the differential detergent extraction kits?
In the case of removing detergents, I guess the tried and true way that even many core labs prefer is to run your samples into a gel and run it in far enough to separate out the detergent and salts from the protein fraction, and then cut out that band containing all your proteins and do in-gel reduction, alkylation, and digestion. But we also found in the case of when we extracted membrane proteins that we could do a methanol chloroform water extraction, and that seemed to remove all of the detergent as well. It's just not as well or predominantly used in core labs, so there might be a little hesitance in accepting that.
Thank you, Barbara. Again, we'll move straight on to the next question that we've had in from one of our users. Somebody's asked, have kits such as MEM-PER been tested on prokaryotes? Would this work efficiently to investigate membrane proteins in bacteria?
Typically, the MEMPER or the membrane extraction kit has been used with mammalian cells and tissues. Prokaryotic cells are a little bit different beast, and we tend to look for other solutions to extracting membrane proteins from those organisms.
Perfect, thank you. And moving on to the next question, are the detergent fractionation and integral membrane protein isolation products applicable to gram-negative bacteria?
No, not in particular.
A nice quick answer there then. Okay, so we'll move on to the next question. Do you recommend combining mechanical and chemical disruption methods for certain cell types or tissues?
Yeah, in fact, you really need to use some kind of mechanical disruption with tissues before proceeding into these types of fractionation strategies. You need to get to the point of where you have a solution of liberated free cells so that you can maximize the surface area for these different buffers and extraction reagents.
Thank you very much. And moving on again, do the antibodies that leach into IP eluates interfere with the downstream MS analysis?
I think I can address that one. I think it depends. So there is some antibody leaching, even with the biotinylated antibody, that does occur during elution from the beads. And for the most part, it does not interfere with protein identification because depending on the instrument that you use, they're pretty sensitive instruments. So although there are antibodies present in the sample, you can still identify your protein of interest. However, if there's too much antibody being liberated during the IP, so for instance with the protein AG IP, it will reduce your ability to quantify. So what happens is that you basically get some suppression of certain peptides, and therefore your lower limit of detection and quantification is impacted by antibody leaching. So therefore, we recommend screening and doing co-IPs is fine for protein AG, but if you want to do quantitative assays, we recommend using a biotinylated antibody to reduce the antibody leaching.
Thank you very much, Ryan. Just to remind everybody in the audience, we have got time for a few more questions, so please keep sending them in. Moving on to the next question that's been asked. Do you have any suggestions or kit availability for membrane protein extraction from plant tissues?
That's a good question. We do have one product. It's actually called PEPR, and it actually uses different solvents to help break down the cell walls of plants, and it does efficiently extract proteins from the plants. However, it doesn't differentiate between membrane versus cytosol, so it is basically positioned as a whole cell plant extraction buffer. But it is one product that we do have that helps liberate proteins for cells, and we have done some mass spectrometry after using those samples through Ingel Digest.
That's great. Thank you, Ryan. As we've got a few questions, I'll keep moving on quickly. Somebody has asked, can a subcellular fraction extracted by differential detergent fractionation be subsequently run through a sucrase gradient for further fractionation?
I don't see why not. That may be a good idea if you absolutely need that very high resolution. I know that once that you fractionate cells, I mean, the endoplasmic reticulum is almost everywhere. So that is very hard to remove, and actually you might get some better separation if you do run a gradient afterwards.
Thanks, Barb. And moving on again, somebody's asked, are the fractionation kits compatible with cross-linking prior to fractionation, such as UV cross-linking?
Yeah, we haven't done that application, but there should be no reason that you couldn't use a cross-linker prior to doing fractionation.
Nice quick answer there. So moving on to the next question that we've had. How does MEMPER plus solubilization buffer compare to SDC for mammalian membrane protein extraction after subcellular fractionation?
So that was to SDC? Yeah.
Yeah,
so
SDC. Sorry, Ryan.
Yeah, so the biggest difference is that the detergent that's in the buffer is non-denaturing, and sodium deoxycholate, or SDC, it is a detergent at high enough concentration can actually do partial denaturation of your protein, and therefore some of the applications that Barb described where we were able to do co-immunoprecipitation, we were able to effectively maintain protein-protein interactions. And secondly, with a strong denaturing detergent like SDC, although it's really good at, I would say, solubilizing these integral membrane proteins, unfortunately, you do also solubilize a lot of other proteins in other cellular compartments, such as the cytosol and nucleus. So I think from a fractionation point of view, it may not be as efficient as the MEMPER Plus formulation. However, as Barb described, there are ways to enhance getting some of these really tough transmembrane proteins extracted. And so therefore, maybe after doing the fractionation, you could use SDC-based detergents to extract more proteins out of the pellets. It just may not be as specific as the detergent that's used in the product.
Yeah, it just depends upon the level of the enrichment and your preference for what proteins you leave out of that fraction. So
Right.
if you don't mind solubilizing everything, then you could solubilize the whole cell in a very denaturing detergent and have success.
Thank you very much. The next question that we've had is, can the antibodies used for IPs be combined for multiplex IPs, and what is the upper limit for multiplexing?
Yeah, so we've actually been working on this, and we do have an immunoprecipitation kit. And most of the data that Barb showed today was a single-plex IP, meaning where we use one antibody per target in a single IP, and we're doing separate mass spec analyses. You can actually do a multiplex IP where you would actually combine your antibodies, 10 to 12 different antibodies together and do multiplex IP, so you can actually monitor more than one protein in a particular pathway. Now, you wouldn't do this for necessarily looking at co-immunoprecipitation reactions because you would basically not understand which binding partner came from which IP. But from a quantitative pathway analysis point of view, it's a really powerful method. And right now, we've actually in our lab done multiplexing up to 12 targets in a single IP. But really what you're limited to there is the capacity of the bead and how many antibodies you can actually pull down. The great thing about doing multiplexed IPs with mass spectrometry versus other immuno platforms such as ELISA or Luminex is that we don't have to worry so much about cross-reactivity because there's no secondary antibodies used for detection. The mass spectrometer is used to basically identify and quantify the proteins. So you can actually multiplex a lot more antibodies that may not be compatible with other immunoassays.
Thanks, Ryan. That's really good. The next question that we've had is, what type of instrument do you need for these experiments? Can a core lab help me with such an experiment?
Yeah, so for the data that we showed today, we actually, most of the data was acquired on two different LCMS systems. One was a Velos Pro ion trap, and that was used for the membrane extraction studies. And the other instrument that we used was the Thermo Fisher Scientific Orbitrap Fusion instrument. And so this is a very high-end, high-resolution mass spectrometer. And what these more high-resolution mass spectrometers are able to give you is many, many more protein identifications. So they're very fast, and they also can deal with more complex samples. So most definitely, if your core facility has access to high-resolution instruments, that does enable getting more protein identifications out of your sample, and you actually get higher confidence in those identifications, as well as the capability of doing quantitation using SILAC or the Tandem mass tags. So yes, so depending on the equipment your core lab may have, some of the technologies we described, obviously all of the protein preparation technologies would be accessible for protein identification. But in terms of quantification, some of the instruments that are available are better at quantifying proteins using SILAC or TMT than others. And so I think it just depends on what equipment you have available.
Thank you very much, Ryan. Unfortunately, that's all we have time for today. But just to remind everybody, any questions that we didn't have time to answer, they will be answered offline as soon as possible. And you can continue to ask questions offline and through the on-demand system. The webinar will be made available on demand now. So if you have any friends or colleagues that you think may be interested in this webinar or any future events, please feel free to share the link. And if you'd like to watch the webinar again and send any questions in that you might have after watching again, please do that. Thanks again for your time, everybody.
All right, thank you.